Hello, welcome to the physiology of the digestive system. There are a lot of chapters to read, and these chapters that are listed are from Guyton and Hall. And Guyton and Hall separates the physiology of the digestive system into things like the kinds of mechanical digestion will be covered in one chapter, and the kinds of digestive secretions will be covered in another chapter. And then in another chapter might be the kinds of hormones that are released, and then another chapter might be the kinds of nutrients you can absorb. So I'm going to try and combine all of these chapters at once as we go from the mouth down to the anus. And we're going to just take one big trip through the alimentary canal. So as I said, we're going to take one big trip. I'm going to first start telling you about the functions of the digestive system. We'll review the anatomy of the GI tract. I'll tell you about the innervation, which is fascinating. Then we'll talk about how there's communication and control throughout the gut, and a lot of this depends on what is physically contacting your gut, like what did you eat, because there's a lot of signals that are released based on what you eat. And then we'll discuss the kinds of mechanical digestion. Those are the physical forces like chewing, aka mastication, peristalsis, you get the idea. And how those help with mixing the digestive enzymes in the gut so that you can properly digest your material. So that goes along with the secretions of the GI tract. And I will, again, talk about the chemical digestion, what's occurring where, and what kinds of absorption you can have in the GI tract. I shouldn't say reabsorption because you are absorbing them for the first time. And then lastly, GI disorders. The whole digestive process involves first with you ingesting material, putting something in your oral cavity for the sake of swallowing it. And the digestive process doesn't end there. There are many digestive processes. You need to be able to move the food once you swallow it throughout the whole digestive tract. You need propulsion. And different areas of the gut are capable of different kinds of propulsion. Peristalsis, pendular motion, churning, etc. And a lot of this propulsion movements can also be involved in mechanical digestion. And the more we mechanically digest our material, the smaller we have our bits and pieces that we've just eaten. And the chemical secretions that we have, the digestive enzymes, can then better attack the chemical bonds in the polymers that we've just eaten. We can't absorb polymers. We can't absorb macromolecules. We must be able to digest them down to their monomers in order to absorb the nutrients in water. And whatever we don't absorb will come out as feces. That's defecation. Let's review the organization of the GI tract, or the layers of the alimentary canal. These you should have learned in anatomy, but I think it's warranted to review them. If we move from the most external layer to the most internal layer, and that means the layer that's facing the lumen or hollow space where your food or feces might be placed, we're going to start with the serosa then and move internal. The serosa really is the visceral peritoneum. Some layers, or I should say sections of the GI tract, don't have a visceral peritoneum. They are considered to be retroperitoneal, meaning behind the peritoneal cavity. And the duodenum is the best example of that. But let's not worry about that. That's part of anatomy. Let's just keep moving internal. As we move deeper from the serosa towards the lumen, we get to the muscularis externa. And the muscularis externa actually consists of two different layers of smooth muscle. There's the outer longitudinal muscle and an inner circular muscle. In between the two, we find a net of nervous tissue. Literally, it looks like a fisherman's net. It's quite beautiful to look at it under the microscope. And this is called the myenteric nerve plexus, or Aurobox nerve plexus. Then as we move deeper towards the lumen past the circular muscle layer, we get to the submucosa. And in the submucosa, we're going to find glands whose job it is to release bicarbonate-rich mucus to help maybe neutralize the stomach acids. The mucus also helps the chyme and or fecal material to actually move along throughout the alimentary canal. And in the submucosal layer, we're going to find another nerve plexus, kind of a net-like meshwork of nervous tissue, neurons. And that's called the submucosal nerve plexus, also called the submucosal nerve plexus. Now I want you to know that both the myenteric nerve plexus and the submucosal nerve plexus together make the enteric nervous system, meaning within your GI tract. These neurons are fully contained in your GI tract. There are as many of them there as there are in your brain and spinal cord. And they can take care of business. They don't need any influence in order to digest your food and absorb it. But we will see in the next few slides that extrinsic nervous systems like the autonomic nervous system will influence these intrinsic nerve plexi. To be continued. Then we get to the mucosal layer, which actually consists of 3 different layers. We get to the muscularis mucosae. This is a band of smooth muscle that can only cause local contraction. I like to think of it as kind of flicking the food that might get stuck in the epithelial lining back into the lumen so that you can have appropriate digestion occur. Then there's a lamina propria, and that just simply means that you have a layer of connective tissue, maybe carbohydrates, that help anchor the epithelial lining to the deeper connective tissue. And then lastly, the epithelial lining. The epithelial lining is stratified squamous epithelium in the mouth and the esophagus, but when you get to the stomach, it turns into simple columnar epithelium. And it stays that way until basically you get towards the rectum that transitions into the anus. We need stratified epithelium in areas where there's a lot of friction, where a lot of the epithelium could be damaged, say from mastication or defecation. But from the stomach throughout most of the rectum, ahead of the pectinate line, as we say, is simple columnar epithelium, which is really key for proper digestion, mixing, and absorption of nutrients. Let's talk about the nervous control of the gut. We have in the submucosal plexus and myenteric plexus a net of neurons that are fully contained in the GI tract. And this is called the enteric nervous system. The myenteric plexus controls the GI motility. It controls the muscularis externa, both the longitudinal layer and the circular layer. And it's involved or responds to stimuli. If there is, say, food stuff that is contacting your mucosa and irritating it, we can actually see chemical signals that will increase the amount of contraction or tone of the muscularis externi. And this will in turn increase the frequency of contraction, and that means you get more propulsion. You get stuff out of your gut. If it's irritating you, get it out. We equate this, of course, to diarrhea. If we get food that's contacting the mucosa and it's okay, it's agreeable to us, we get the submucosal plexus releasing secretions and participating in chemical digestion, which in turn leads to absorption. And again, the muscularis mucosa, it helps with this by flicking the food back into the lumen so that you get proper mixing. Now I want you to know that this intrinsic neuronal system is its own system. There are sensory neurons that monitor tension and what kind of food you have in there, and there's also efferent outgoing commands. Should we increase propulsion, get rid of this, it's nasty, more diarrhea? should we absorb it? And all of this can function on its own. However, as seen in this picture, you can have influences, if you look at the bottom, from both the parasympathetic division and the sympathetic. So there is extrinsic involvement, but these extrinsic nervous systems can only augment or facilitate change. Really, the enteric nervous system can handle business on its own. It has its own kinds of mechanoreceptors and chemoreceptors. It has its own endocrine cells to release hormones to help with digestion and gut motility. And it has its own secretory cells. So the gut can function on its own. It has its own reflex arc, all the 5 components, a sensory component, afferent fibers, integration, except in this case it's not in the brain or spinal cord, it's totally within the gut, and then outgoing commands and effectors. And the effectors might be the smooth muscle, or the muscularis externa, or muscularis mucosae, or it might be digestive glands. Truly a remarkable system. Just because it's so remarkable, I want to give you another example. Down at the bottom picture, you're seeing the myenteric plexus and the submucosal plexus. And these are fully 100% contained in the alimentary canal. And I want you to see, if you read the picture carefully, these neurons can send sensory information up to higher levels of the body, the spinal cord, and even to the brain. And as such, also the brain and spinal cord can send output information through the sympathetic and parasympathetic divisions. And if you read on the left side of the screen, the parasympathetic and sympathetic divisions participate with communication of the gut through relaying both afferent, that means outgoing, relaying sensory information, and then bringing back down commands, that means efferent. I'm going to say efferent so you can hear the difference between the words. Do not memorize the percentages of afferent versus efferent, or afferent versus efferent. Just know that there is extrinsic influence into the enteric nervous system. Now, how can we relay what's happening in the gut to other places of the body, even to other areas of the gut, and even to the brain and spinal cord? Well, it comes down to local factors, neural mechanisms, and hormonal. And I told you at the beginning of the semester, we're going to see this theme often. There's local control. Let the organs take care of business on their own. If things go awry, then let other neural systems and endocrine systems come in to lend a helping hand. Neural mechanisms will be quick to respond, but quick to shut down. Hormonal mechanisms will be slower on the uptake, but they will last longer. So if you look at the bottom left picture, you can see local factors can actually stimulate the mucosae and submucosae. And that can ripple out from the submucosal plexus out to the myenteric plexus. Local factors like there are sensory cells that are monitoring the pH, chemoreceptors. They're monitoring the physical stimulation of the gut. Is there food there? Is it pressing on the gut? And even chemical stimulation. What kind of food did you eat? Is it not just acidic, but does it have some caspacin? Did you eat something spicy? Or did you eat something where it's rancid? So it's not just pH, but other chemicals that your gut is able to detect. And as you follow the picture upward, you can see that there are stretch receptors in addition to chemoreceptors, and all of this is communicated to the submucosal plexus, the myenteric plexus, and even up to the brain for higher processing. But locally, if something, quote, disagrees with your gut, and that's a real thing, there will be increased gut motility to get that out. And that means you're either going to vomit or you're going to have diarrhea. In addition, we can see that even the local signals can send information to enteroendocrine cells. That's a fancy way of saying cells within the gut that behave in an endocrine fashion, which means they're going to release hormones into the blood. And even though these hormones are released into the blood, they can still circulate back and target other centers of your gut and change their physiology. If you look to the right, there are clearly gastrointestinal proteins that can act as paracrines. These paracrines can be released from cells and target neighboring nearby cells. That's what a paracrine means. Crine means signal, para means next to. So signal the cells next to me. Are we in distress? Then you should be on board too, neighboring cell. Neurocrines, that's a fancy way of saying neurotransmitters. And we will review the neurotransmitters that target the gut. And then on the left side, we see hormones. And definitely, yes, there are a lot of hormones that will be released that actually go into systemic circulation, only to come back and target cells within the gut, or in the case of insulin and glucagon, for example, can target other cells of the body, like insulin-dependent cells or the liver. So let's talk about some hormones of the gut. And you should know these hormones that I'm listing and where they're secreted and what makes them secreted in the first place. What are the stimuli and what will they do? Now gastrin, and I will say this again later, is secreted by enteroendocrine cells called G cells, and they are found in the stomach. And gastrin responds to peptides or amino acids physically touching your stomach. And when gastrin is released, it leads to even more hydrochloric acid secretion by the stomach. Cholecystokinin, or CCK, is released by enteroendocrine cells found in the duodenum, jejunum, and ileum. These are called I cells. And notice that CCK is released again because of protein or fats and acids. And CCK leads to more pancreatic enzyme secretion, digestive enzymes. And it also slows down gastric emptying. This should make sense because you want time to kind of churn the food and mix it with the digestive enzymes. CCK also causes the gallbladder to contract, which means bile will go through the bile duct and into the duodenum. Secretin, I think S for secretin, S for S cells of the duodenum, and S for slippery bicarbonate-rich buffer. Secretin is going to help neutralize gastric acids, and it will decrease gastric secretion of hydrochloric acid. And notice that secretin is released from acids or fats physically touching your duodenum. Lastly, there's a hormone called GIP. Its original name was gastric inhibitory peptide, or GIP. Since then, it's been reintroduced as glucose-dependent insulinotropic peptide. It's released by the K cells of the duodenum and jejunum. Notice the physical presence of protein, fat, or carbohydrate stimulates its release. When GIP is released, it stimulates insulin secretion. And I'm going to talk about this in a little bit when I review diabetes mellitus, because I will share with you that my dietitian told me that it wasn't just important to watch my carbs, but it was also important to eat my food in a certain way. She wanted me to eat my proteins and fats first. When the proteins and fats physically touched my gut, this would lead to GIP release. And before I even ate the carbohydrates, the GIP would in turn stimulate insulin secretion from the pancreas. So my insulin-dependent cells would already be primed with their glucose transporters before I even ate my carbohydrate. So I do not want you to memorize at all the stimuli for secretion. We can summarize this entire column as the physical contact of food you just ate causes these enteroendocrine cells to release their hormones. The physical contact of food on the mucosa. And I find that to be truly remarkable. Some paracrines. This means chemical signals that work on local cells. 2 I really want you to know. Histamine. Histamine is typically thought of as a distress signal involved in the inflammatory response. It is released by enteroendocrine cells in the GI tract, not just in other places of your body. Specifically and hugely in the stomach, where there's a lot of acid release and a lot of irritation that can occur. Read into that ulcer development. So when histamine's released, ironically, even though it's a distress signal and can be released by the cells of the stomach when exposed to too much acid, this is going to have a feed-forward effect, and it's really bad. It's going to stimulate even more hydrochloric acid secretion from the parietal cells. Another paracrine is somatostatin. This is technically both a paracrine and a hormone. We've seen somatostatin earlier in Unit 1 when we learned about growth hormone and inhibitory hormone. Somatostatin is released by cells in the GI tract, but also by cells in the hypothalamus and also cells of the pancreas. All I want you to know about somatostatin is that it inhibits secretion of GI hormones and inhibits gastric hydrochloric secretion. In some, it merely slows down the digestive process to make sure that we don't get ahead of ourselves, so that we don't over-digest and maybe start digesting our own mucosa and leads to ulcers. So it just kind of slows things down to make sure we neutralize acids appropriately and make sure we chemically digest things appropriately and absorb them. Slow and steady wins the race, is what I like to say. Neurocrines, aka neurotransmitters. If you're reading Guyton and Hall, there are actually quite a few, and I've listed some of the ones that Guyton and Hall talks about here. Some of the ones listed in black are actually painkillers, and some actually detect pain in the first place. But the 2 I want you to know are listed here in red. Now, can you guess why I picked just these 2? You should recognize by now that these 2 neurotransmitters represent either the parasympathetic division or sympathetic. Parasympathetic acetylcholine increases gut motility. Epinephrine/norepinephrine leads to decreased activity of the gut, reduced gut motility. Let's talk about motility for a little bit. This is so awesome. Now, you're going to learn about some cells that can depolarize on their own without a neuron telling them to do so. Not just in the gut, but in your heart as well. The cells in your gut that can depolarize on their own, look at the right picture, letter A. These are called the interstitial cells of Cajal. They're considered to be the pacemakers of the gut. They, throughout time, throughout the next few minutes or so, they will spontaneously depolarize and then repolarize and then depolarize again and repolarize. And we've learned that if they reach threshold, when depolarization reaches threshold, then you can have an action potential. And the light purple lines occurring on the top of the depolarization wave are bursts of action potentials, which means they occur, we come back, they occur again, and you have about 3 to 12 waves of these depolarizing events per minute. Again, it's autorhythmic. That means these cells are able to depolarize on their own. They have fickle membrane potentials. And once an action potential is started, it ripples throughout the gut because all of the gut cells are connected with each other through gap junctions. I think that's really amazing. And I'm talking about the muscularis externa because we're talking about motility, so we need muscle. So all these smooth muscle cells are connected by pores in their cell membranes, specialized connections called gap junctions. And then in the bottom picture where it says B, you're seeing In yellow dots, the mechanical event that occurs due to the electrical events. The gut starts to contract, and that means you get gut motility. Our last slide of the first installment of this lecture continues with motility. When we swallow, or deglutition, we are basically relying on contraction, as shown here in A, number 2. We're seeing a contractile ring, and then on either side we're seeing relaxation. When we actually swallow and it leads to peristaltic contraction, we see a contractile ring behind the bolus, but relaxation ahead of it, and this is called receptive relaxation. And once we get to the esophagus and beyond, this now becomes the law of the gut. Contract behind the bolus, relax ahead of it. That basically means food in and feces out. So that's good. We're going to see in letter A, just go back to that, letter A does not represent swallowing, and I misspoke if I did. Doesn't represent deglutition. It represents segmentation, or a local contraction. The segmentation occurs in the small intestine, where it's providing local contraction to help mechanically disrupt the food, and I'll talk about that some more. In the stomach, we see yet another kind of mechanical breakdown. This is called retropulsion, or churning, where the stomach basically flips the food like a chef flipping a pancake over and over. And this helps with not just mechanical digestion, but also chemical. We're going to see that the cells of the stomach, the chief cells, release an enzyme involved in protein digestion. When the stomach starts this process, the food you ate turns into about 1.5 liters worth of fluid, of chyme. Once you mix your food with the stomach secretions, it's called chyme. And after a meal, you can have about 1.5 liters to 2 liters worth of chyme. And it will take about 3 hours for a full stomach to completely empty into the small intestine.